We so often focus on what a new drug does, right? Its big therapeutic effect. But the how that drug actually navigates the incredibly complex landscape of our body to get to its target, well, that's an equally fascinating scientific story. And that's the science of drug delivery. So today we're going to break down exactly how that journey works, all the way from the moment a pill is swallowed to that final interaction at a molecular level. Let's get right into it.
So here's our game plan for today. We're going to start with the big picture, the three key factors that really govern all of drug delivery. Then, we'll zoom right in on what is probably the single biggest hurdle for most medicines, crossing the intestinal barrier. From there, we'll get into the real nitty-gritty, the chemistry of absorption itself. After that, we'll see how a drug's physical formulation, the pill itself, plays a huge role. And finally, we'll look at some absolutely incredible, cutting-edge strategies that are changing the whole game. Okay, first up, let's set the stage and look at the big picture. It turns out there are three main overarching factors that decide whether a really promising drug molecule can actually make it in the body.
And just look at the sheer scale of this challenge. It's mind-boggling. Researchers might start out with something like 10 or even 20,000 potential compounds. After years and years of research and clinical trials, that massive number gets funneled all the way down to just one approved drug. And a huge reason for this massive drop-off isn't that the compounds don't work in a test tube, it's delivery. I mean, a molecule can be incredibly potent in a lab, but if it can't survive the journey through the body to its target, it is completely and utterly useless.
So successful delivery really rests on these three pillars. First, the drug's own molecular structure. This is pure medicinal chemistry. The actual shape and properties of the molecule itself are what dictates its solubility, its stability, and how it interacts with its target. Second, you've got product formulation. This is all about pharmaceutics, how the drug is physically put together, whether it's a tablet, a cream, or an injection. That formulation controls how the drug gets released. And third, the route of administration. This is where pharmacokinetics comes in, the study of how the body affects the drug. The route you choose, oral, IV, topical, it determines the drug's entire journey of absorption, distribution, metabolism, and finally, its elimination from the body.
Okay, so we've seen the big picture. Now let's zoom right into the cellular level and focus on the single biggest obstacle for most oral drugs, crossing that intestinal barrier.
For any drug you take by mouth, the intestinal epithelium is the critical barrier it has to get past to reach the bloodstream. This diagram here shows the most common route it takes, which is called the transcellular pathway. So a drug molecule first has to get into the cell membrane on the apical side. That's the side facing the inside of your intestine. Then it has to travel all the way through the cell and exit through the basolateral membrane on the other side to finally reach the blood. And this whole process is typically what we call passive simple diffusion. That just means it doesn't need any energy, and it simply follows a concentration gradient, moving from an area of high concentration to low concentration. As we're about to see, this pathway really favors small, fat-loving, or lipophilic compounds.
So let's just make sure we're clear on those terms. Lipophilic literally means fat-loving. These substances dissolve really well in lipids and fats. On the other hand, hydrophilic means water-loving, and these dissolve well in water. The fundamental principle at play here is like dissolves like. And since our cell membranes are basically a lipid bilayer, a fatty sandwich, lipophilic molecules are the ones that can get through. Hydrophilic molecules, well, they really struggle to cross that barrier. But here's the catch. It's a very delicate balancing act. A drug absolutely needs to be lipophilic enough to get into the membrane in the first place, but if it's too lipophilic, it can literally get trapped inside. It just loves that fatty environment of the membrane so much that it never wants to leave, so it fails to exit the other side and never reaches the bloodstream.
All right, now that we understand this whole concept of lipophilicity, let's dive even deeper. Let's look at the specific chemical properties that allow scientists to actually quantify and predict a drug's ability to cross these biological membranes.
So how do we actually measure this property? Well, the key metric that everybody uses is called log P. It's just a single number that quantifies a drug's lipophilicity. It's calculated from something called the partition coefficient, which we just call P, and that simply measures how a drug decides to distribute itself between a lipid-like solvent and plain old water. And the way they measure this is with this surprisingly powerful and pretty simple method called the shake flask experiment. You literally take the drug and shake it up in a container with a mixture of octanol and water, which don't mix. Now, octanol is a lipophilic solvent that acts as a great stand-in for the lipid bilayer of a cell membrane, and the water, of course, represents the watery environment like our bodily fluids.
After a good shake, you just let the two layers settle out, and then you measure the concentration of the drug in each layer. Simple as that.
So here's how the math works out. First, we find that partition coefficient, P, by dividing the drug concentration in the octanol by the concentration in the water. So in this example, we have 0.8 divided by 0.2, which gives us a p-value of 4. Now, because these p-values can have a massive range, we convert it to a logarithmic scale to make the numbers easier to handle. So we take the base 10 log of p, and that gives us our final log p. The log of 4 is 0.602. And that's it. That's our log p-value.
Now, log P is absolutely essential, but it's not the whole story. And that's because many drugs can gain or lose a charge. They can become what we call ionized. And this is where pKa comes in. A drug's pKa tells us the exact pH at which it is perfectly balanced, 50% ionized and 50% unionized. And here's the critical takeaway. Those charged, ionized molecules are way less lipophilic, and they really struggle to cross cell membranes.
So how do we connect a drug's pKa to its actual environment in the body? Well, scientists use the Henderson-Hasselbalch equations. You can think of these as the mathematical tools that allow us to predict exactly what percentage of a drug will be in its absorbable unionized form at any given pH. This is absolutely critical for knowing whether a drug is going to be absorbed better in the superacidic stomach or the more neutral intestine. This all leads to an incredibly useful clinical rule of thumb. When the pH of the environment is just one single unit away from the drug's pKa, the drug is already about 91% in either its ionized state or its unionized state. This just shows you how incredibly sensitive absorption is to pH. A tiny little shift can completely flip a drug from being almost totally absorbable to almost totally non-absorbable.
All right, we have gone super deep into the chemistry of the drug molecule itself. Let's now pull back a bit for our next section and see how the drug is actually prepared and packaged into a final product, and how that affects its journey into our bloodstream.
Now, despite all those absorption challenges we just talked about, oral dosage forms like tablets and capsules are still by far the most preferred way to take medicine. And the reasons are pretty clear, right? They can be self-administered, which is incredibly convenient for patients. They're generally pretty inexpensive to manufacture. Absorption can happen along the entire length of the GI tract, and this format allows for the creation of all sorts of advanced controlled-release formulations.
You see, a tablet isn't just a compressed puck of pure drug. It actually has two main components. The first is the active pharmaceutical ingredient, or API. This is the drug molecule itself, the thing that produces the therapeutic effect. But it's not just its chemical identity that matters. Its physical form, whether it's a crystal or an amorphous powder, and its particle size can seriously affect how it gets absorbed. The second component is the excipients. These are all the supposedly inactive substances, things like fillers, bulking agents, and lubricants, that are added to help with manufacturing and to control the drug's release. This brings us to a really vital concept, bioequivalence. How do we know for sure that a generic drug works exactly the same as the original brand name one? Well, the gold standard test is something called a crossover study. Here's how it works. You get two groups of volunteers. One group takes the brand name drug, let's call it product A, while the other group takes the generic, product B. Then, after a washout period to make sure the drug is completely out of their systems, they switch. The group that took A now takes B, and vice versa. Throughout the entire study, blood samples are taken over and over to measure the drug concentration. If both products produce the same concentration profile in the blood, they are considered bioequivalent.
So what can actually affect absorption at this stage? The factors really fall into two main camps. First, you have pharmaceutical factors. These include tiny variations in the manufacturing process or differences in those inactive excipients we talked about. Second, you have physicochemical factors, which are all about the drug itself. This includes its salt form, its log P, its particle size, remember, smaller particles dissolve faster, and even its degree of hydration, which is just how many water molecules are stuck to.
Okay, so we've covered the fundamentals and we've covered the challenges. Now for the really exciting part. Let's wrap this up by exploring some cutting-edge strategies that are designed to blow past these hurdles and target drugs to specific sites in the body with incredible precision.
Here is a brilliant strategy called passive targeting that cleverly takes advantage of a tumor's unique environment. Here's the breakdown. The cause? Solid tumors often grow so fast, they actually outgrow their blood supply, which means they're very low in oxygen or hypoxic. The mechanism? Scientists create what's called a prodrug, basically an inactive molecule. This prodrug has a special chemical group on it, a nitro group.
In the low-oxygen tumor, that group gets chemically changed into an active DNA-damaging agent. And the consequence, the drug is only switched on inside the tumor, where it goes on to attack the DNA of the cancer cells, leaving healthy, oxygen-rich tissue totally unharmed.
Let's look at two more really advanced strategies. First is active targeting with antibody conjugates. Okay, think of this like a molecular guided missile. A superpotent drug is attached to an antibody that is specifically designed to seek out and lock onto a protein found only on cancer cells. The drug can be attached with a special chemical bond that's stable in the blood but breaks apart in the slightly acidic environment of a tumor, releasing the drug right where it needs to be. Second is a clever chemical trick called bioisosterism, using deuterium analogs. Here, you just replace a regular hydrogen atom with its heavier, non-radioactive cousin, deuterium, at a specific spot where the drug normally gets broken down by the body. A carbon-deuterium bond is stronger than a carbon-hydrogen bond, which slows down the rate of metabolism and improves the drug's overall profile.
So from the basic physics of passive diffusion all the way to these incredible molecular guided missiles, it is just so clear that the future of medicine isn't just about discovering new drugs. It's about inventing new and better ways to deliver them with more and more precision. And it leaves you with a final thought, right? Having figured out how to tackle cell membranes, pH, even oxygen levels, what barrier will we learn to overcome next?